So I guess, one of the things and one of the big black boxes in this field is how could potentially bacteria in the gut signal to the brain? Like, what are the potential factors involved? And it's still a black box somewhat. One of the ways is through a disrupted barrier function. I'll talk a little bit about that. And we know in aging that barrier function becomes, the barrier becomes much more permeable, and allowing bacteria to infiltrate, allowing a pro-inflammatory state to occur. But also we know that the direct effects on the immune system, on the HPA axis, tryptophan metabolism, we're getting a lot of evidence which can also have immunomodulatory effects. But it's worth noting that bacteria can produce short-chain fatty acids, like butyrate. And butyrate is a very well-known histone deacetylase inhibitor. So it can have epigenetic effects. Bacteria can produce all types of neurotransmitters and neuroactives, and they're really little factories pumping out all sorts of novel substances depending on the substrates that they have. And this is just some examples of that. Work from colleagues in Cork has shown that you can select a bacteria that produces the neurotransmitter GABA. Also, you can get bacteria that will change the composition of fatty acids in the brain, which may have a positive effect on things like cognitive performance. Work from Elaine Chow and Sarkis Mazmanian at Caltech earlier this year has shown that you can select for sponge-forming bacteria that will modulate the serotonin system locally in the gut and really change how serotonin has an effect on motility and other readouts. And indeed, Mark Light, who's now at Iowa, has pointed out that most of the neurotransmitters, but also even some novel neuroactives that we haven't even thought about or even discovered properly, have the potential to be produced by these bacteria. And it is these metabolites that may be driving some of the effects, both locally in the gut, or even some of them may even signal to the brain. And so slowly we are trying to disentangle what are the microbes doing on locally at the gut in terms of the immune cells, in terms of the releasing of gut hormones and neuropeptides from enteroendocrine cells, how they're impacting on the enteric nervous system, the second brain as we call it, and how that can influence brain function at multiple levels. A lot of work to be done, a lot of exciting areas to pursue, and it's a real interface of multiple disciplines from metabolism, neurogastroenterology, and neuroscience, as well as microbiology and big data as well. So how do we study the microbiota gut-brain axis in my lab? Well, there are different ways that we do this. I've mentioned probiotics already, and I'll show you some data with probiotics. I showed you the antibiotic data we have, which is also another way for looking at this. Infection studies have been the founding basis of psychoneuroimmunology as a discipline. Germ-free studies. These are mice or rats that grow up in a completely sterile environment. Now, most of us who run mice experiments in SPF, we think they're very clean and they're very sterile, but germ-free is this at an extreme, and you need very dedicated notobiotic facilities to do these studies, and we are lucky to have one in Cork. And then finally, there's fecal transplantation studies, which is exactly what it says on the tin, and I'll come back to that. So the germ-free concept has really, taken the imagination of people for almost 100 years. You can see it here. This is a germ-free man in this sci-fi magazine from 1927. And the first animals emerged in the 1940s, and people started studying them. And in particular, a lot of emphasis in the 1960s and 50s, so this is from 1958 and 1966, were showing the role of the microbiome in the aging process. And if you could keep a mouse in a germ-free facility, it lived longer. It didn't have as much fun, but it lived longer. So, it's always a risk-benefit factor. It's maybe a bit like some of the nuns that live in enclosed convents who live longer, but they may not have as much fun. So these mice have been really brilliant tools to answer very specific questions. Is the microbiome involved, yes or no? They don't really have a human correlate. The boy in the bubble would be one potential correlate, but on the whole, they help us just assess very clearly some very specific questions. And there's a paper over a decade ago from Sudo's group in Japan, which showed if you took these germ-free mice and stressed them, they have an exaggerated stress response in terms of the hormonal outputs. And moreover, if you put back in a single bacteria, a bifidobacteria, which is present in many probiotics, you could reverse this exaggerated stress response, both at ACTH and cork levels. And this paper was published in the JPhysiol, but it didn't gain a lot of attention at the time. It took people a while, and it was about 2 years later before I actually paid much attention to it. And then you start thinking, well, if the stress response is out of kilter in the absence of microbiome. And we then had shown, as I showed you earlier, that stress will affect the composition of the microbiome. It starts getting you thinking about what are the neural basis of this, and what's going on in the brains of these animals. And so as a neuroscientist, we were quite interested, does it change neurodevelopment? And like many great things in science, just when you come up with an idea, two other groups come up with the same idea at the exact same time and publish it before you. But that's okay, because what's really nice about these studies is they all show the same thing, which in behavioral neuroscience is also quite nice to see, using different strains and different apparatuses. And so work from Rochelis Heitz and Sven Pedersen at Karolinska, and Jane Foster's group, and Karen Ann Neufeld, who's now a postdoc in my lab, they showed that if you grew up germ-free, that your brain development is abnormal and that you have a change in anxiety-like behavior. So we showed the same, except one thing that we noted was this seemed to be sex-dependent, and that most of the effects were more prevalent in males, not females. Now, if you're a stress researcher, you tend to focus on outputs that are more prevalent in the female population. And the NIH is very pushing this idea of looking at both sexes now in neuroscience. And so this got us a bit perplexed, and I'll tell you where that led us in a few minutes. So here's some data just showing in germ-free animals they have an exaggerated corticosteroid response like Sudo had shown before. When we looked at the hippocampus and we looked at brain-derived neurotrophic factor, a key plasticity-related protein involved in learning and memory and the antidepressant response, this was decreased in the hippocampus. Serotonin levels were increased. And so we found wide-scale changes also in gene expression in these animals. Another process that we've been interested in for many years has been the process of adult hippocampal neurogenesis. These are the cells in the hippocampus that continue to be born throughout your lifespan. And we know for many years that factors such as stress and inflammation and aging will dampen down these or decrease the number of new cells being born, whereas learning and enrichment and exercise, as well as antidepressant drugs following chronic administration, will increase these new neurons being born. And so Olivia O'Leary, a colleague of mine in Cork, and I were interested in whether the microbiome could play a role in shaping how these new cells are formed. And so we looked at both survival and proliferation and neurogenesis, as we call it, in the hippocampus of these animals. And we find a clear change and actually an increased neurogenesis in animals that didn't have a microbiome, and it made the cover of Biological Psychiatry in August, telling us that how the break on how neurogenesis is regulated is dependent on signals from the gut, which is quite interesting. More recently, more data is unpublished, but it's just to make sure that we're not just hippocampal focused. Another brain area we're very interested in is the amygdala, which is very involved in fear and anxiety. And we find in germ-free animals there's an increased amygdala volume, an increased architecture of amygdala neurons in terms of here at the BLA level, and in terms of spines, that there's an exaggerated increase in spine density, and this is what's very important for neural connections. When we look at gene expression using deep sequencing technology in the amygdala, we find that there are clear genes that are involved in immediate early gene activation. And so in the amygdala, there are clear differences going on in terms of how the amygdala responds to a stimulus, and in germ-free versus control. So it got us really thinking about, functionally, how can the microbiome shape both hippocampal, amygdala, and probably also some cortical pathways. Last year, a very important and interesting paper emerged from Sven Pettersson's group in Karolinska, which showed that the blood-brain barrier permeability is also regulated by the microbiome. Here you can see animals that have normal animals, and this is animals that are germ-free. And you can see as a radio tracer, which is now able to get into the brain. And very intriguingly, they showed that if you gave a specific potential probiotic to these animals, that you could recover this. And these probiotics made butyrate and other short-chain fatty acids. So the short-chain fatty acid production was able to tighten up the barrier again. And so this is also quite important as we age, because we know blood-brain barrier permeability increases in aging and in Alzheimer's disease. And then there still was, when I would speak to neuroscientists, there still is a certain level of healthy skepticism in the field regarding the influence of the microbiome on brain function. And again, it goes back to this below-the-neck type scenario. But then a paper emerged this summer, which I think really got to get a lot of people thinking in areas they hadn't thought before, and that's about how microglia are regulated. And this is work from Marco Prinz's group in Germany, which basically showed that using germ-free animals and other strategies, including antibiotics, that how these microglia become activated will play a key role, which plays a key role in neuroinflammatory processes, and we know is exaggerated in aging and Alzheimer's disease, etc. And that this is under the control of the gut microbiome. And it's under the control of some of the short-chain fatty acids produced by the gut microbiome. And so it tells us that potentially we can regulate the microglia in our brain by targeting microbes in our gut, which is very much a new way of thinking. And microglia are the macrophages of the brain. So which brings me back to our old friend Mechnikov, who discovered the macrophage. And in this commentary, we wanted to highlight, some of the convergence that brings to the field, because in the aging and neurodegenerative disease, we do have a clear activation of microglia occurring that will cause neuroinflammation, have impact on synaptic pruning, etc. And that there are signals now coming from the periphery to the brain that are targeting this microglia activation. And so this is a process which people have for a long time been using minocycline, which is inhibits microglia activation locally in the brain. But people have forgotten that minocycline is also an antibiotic and will also affect the bacteria composition in the gut, and may need to rethink some of their minocycline studies again overall. And minocycline is ongoing trials in Europe in different psychiatric disorders at the moment.